Scarlet, because I'm falling in love and I am not ready to die. Scarlet fever is killing me.
University professor Dr. Livdahl in his lab. So tell me a little about yourself.
Well, I'm an ecologist and I've been here at Clark since 1980. And I work on species interactions. So the effects that different species have on one another, and these might be predators and prey, or they might be competing species. So that's where I've done most of my work, and we've focused primarily on the mosquitoes that lay their eggs in small containers, which are convenient communities to work with, because we can do work with them in the field, and we can also bring them into the lab and do experiments with them here. So recently we've gotten involved with parasites of mosquitoes, which have some interesting effects on other interactions that might occur between mosquito species. So,
and what classes do you teach at Clark?
I teach ecology and other courses that are quite ecological, like ecology of disease vectors, ecology of infectious disease. I teach an introductory biostatistics course. I co-teach ecology of Atlantic shores. So those are most of the courses that I've been teaching recently.
It's a pretty hefty workload.
Oh, I don't teach them all at once, but and some of them I teach in alternating years.
Tell me about the research being conducted here.
Well, our project is going to focus on mosquitoes and mosquito communities as hosts for parasites. So we're studying the influence of parasites on mosquito success. These are parasites of mosquito larvae. And we're also studying the influence of the diversity of a community of mosquitoes on the success of the parasite. There's an idea in the disease ecology literature called host dilution, which suggests that when a parasite has multiple hosts, multiple species of hosts, that some of those hosts are going to be better for the parasite than others. So by having more species, there will be more possible dead ends for the parasite. So having more species might not be as good for the parasite as having fewer species. So we're interested in testing that idea by constructing different communities of mosquitoes that have different numbers of species in them and seeing how well these parasites do in these different setups.
Interesting. And I understand you just received a hefty $380,000 grant. Do you want to talk about that a little?
Well, yeah, it was given to us to support this project I just described. So, setting up different communities of mosquito hosts and looking at how successful the parasite is in those different situations is what we're going to try to do. We're also going to be going to a lot of places in the eastern U.S. and sampling mosquito communities, container breeding mosquito communities that have different numbers of species in them, just to see if there's any relationship between the prevalence of parasites and the number of species that are present.
Oh. And what do you believe is the potential future applications of this research?
One important thing that it might provide is some evidence that supports the whole idea of biodiversity. That is, preserving species is something a lot of us think that is a good thing to do, but we don't have a lot of practical reasons to do it that have been well demonstrated and well tested. So one thing that could come from this is more justification for conservation efforts. Okay, we do some molecular work in our lab, usually just trying to quantify or to identify species. And so we extract DNA and we use a technique called PCR to amplify DNA and to enable us to identify species, either of parasites or of mosquitoes. And also we're hoping to use that to quantify the amount of parasite material that exists within a mosquito larva or within a mosquito adult. So we do a lot of gel preparation and DNA extraction here.
Okay. How about on the other side of the
mosquito
lab?
larvae. Over here we have microscopes which we use to identify mosquito larvae or eggs, as well as to manipulate them in various ways. For example, we have some mosquitoes that we work with that are difficult to get to produce fertile offspring in the lab, so we force mate some of them by knocking the females out and knocking the males out and putting them together. So we do some of that over here. Various things having to do with processing of field samples goes on over here. So we pick mosquito larvae out of muck like this and count them, and we do a lot of measurement of things with the microscopes too.
Is it difficult to differentiate between the species?
Sometimes, yes. We do work with a couple of species that are pretty difficult to tell apart. We should take a really close look at them. So that's kind of time-consuming and labor-intensive.
I can imagine.
Yeah. And then here we have mosquito colonies growing. We have generally pretty dim lighting in here, but if you walk in here, we have a bunch of different species. We have several different mosquito species. Some of them breed in tree holes, some of them breed in tires, some of them breed in rock holes, and some of them breed in pitcher plants. Several different species going. Some of them need to bite, and some of them don't need to bite at all. Some of them eat other mosquitoes. So there's a variety of different things that we look at.
That's interesting. I actually wasn't aware that there were mosquitoes that didn't need to bite.
Yeah, there are. We have a couple of different mosquitoes here that don't bite. One of them is a predator on other mosquito larvae as a larva, and the other one lives only in pitcher plants. It doesn't bite either.
A mosquito that preys on other mosquitoes. It sounds like
Yeah,
friend of man.
it is. It's a nice species to work with. Kind of difficult to work with too, because we have to provide them with food, which is mosquito larvae, which we have to raise. So you would think that it'd be less trouble to raise a mosquito that doesn't bite, but it has to eat other mosquitoes that do need to be fed. So that's a bit of a problem. But recently, this grant has allowed us to buy a membrane feeder, which allows us to feed the mosquitoes with blood that's heated up across the membrane. So that's a nice new addition to the lab.
It's got to be an exciting treat for
Simplifies
mosquitoes.
the whole feeding process.
Thank you so much for coming on the show.